Fusion.
Hello,
Hi, Ian. How are you?
I'm good, thanks. How are you?
Thanks.
That's good to hear. And you're going to be giving a talk at the Frontiers of Science Forum on Friday about learning from nature to create anions. But what are anions?
Anions. So,
Anions.
so anions are negatively charged atoms or molecules.
Fantastic. So you're going to be creating negatively charged atoms and or molecules by learning from nature. And what are these for?
Okay, so I'm not creating the negatively charged ones. I'm creating things that will bind to those or sense them or extract them. So, um, yes, let me think back to what your original question was.
Well, I'm trying to get to the focus of how you
Yeah.
explain this part of your work.
Yes. So basically, the inspiration from nature really comes from the area of chemistry that I work in, which is something called supramolecular chemistry, which has been defined as chemistry beyond the molecule. But it's really looking at interactions between molecules and how we can try as chemists who make new molecular structures to mimic what nature does. Nature's proteins, enzymes, they do amazing things. They selectively bind to species. They convert them to different things. They self-assemble to form bigger molecular structures that have different functions. And we're not very good at doing that as chemists with smaller molecules. So the area of supramolecular chemistry is really in trying to develop small molecules that have the same functions as those much larger biological molecules, like things like proteins and DNA, for example.
Wow, so that's an enormous field. And so you're working in anything from sensors to medications to new materials, all sorts of things.
Uh, Yeah, absolutely. And so the focus, my focus is on negatively recognizing negatively charged species, so anions. The reason being that they're everywhere. They're in the environment causing pollution. They're in industry, feed streams and waste streams. They need to be removed out of, and they're in every biological reaction. Pretty much involves a negatively charged species at some point. So being able to selectively sense those or remove them from a system or move them from one place to another in a cell or in a waste stream has applications that stretch from the environment to biomedicine and industry. And the fundamental science is the same for all of those applications.
And what natural processes are you examining?
So we use nature has developed a range of proteins that bind to anions with very high selectivity. And so we look at what those proteins do and the interactions they use to bind to their target, whether it's a sulfate ion or a phosphate ion. And then we try and construct much smaller molecules with the same types of interactions that will allow us to bind to a target. And those targets for my research are generally things like sulfate and phosphate and nitrate, but then also biological molecules that contain negative charges as well. So DNA is a big anionic species. ATP is an anion, and so are many of the building blocks that are used, or the intermediates that come out of reactions that use ATP. There are phosphorylated peptides and proteins that are anionic and have signaling, whether they're anionic or not impacts their signaling ability inside cells, so it turns it on or off. And the lipids that make up your cell membrane, many of those are also anionic in nature. So being able to selectively bind to those will give us tools that can help us understand biological processes.
That's a huge range of things there. And of course, ATP is how cells turn food into energy.
Yeah.
And so, what would you be binding to there? Would you be looking at illnesses where it doesn't work properly, or what would be happening?
So in that area, we're mostly focused on looking at binding to either ATP or the products that it's broken down to in enzymatic reactions. So pyrophosphate is one of those intermediates, and phosphate is another. And looking at trying to selectively detect each of those, that will allow us to monitor enzymatic reactions, for example. So if you can selectively detect pyrophosphate, you can actually start to monitor DNA synthesis, amongst other things.
Amazing. So, there's huge applications, and lots of them, the applications are actually also where you're looking at the natural processes.
Yeah, absolutely, absolutely. And sort of the processes, I would say that that's more on the biological side. So the industry and environmental side is really just about sensing those anionic species,
Right.
provide tools that would allow us to monitor concentrations, detect pollution or remove species from water that, has applications across wastewater treatment, water recycling, being able to monitor whether reverse osmosis processes are actually taking all of the nasty things out of our water streams. On the biological side, what we're trying to do is to develop tools that will help biomedical researchers understand biological processes in a better way. So a lot of the time that's around either looking at enzymatic reactions, or it's about looking at in the lipid area, how changes in the composition of a lipid membrane, a cell membrane, impact what's happening in the cell. It's something that we don't really understand very well yet as scientists in general. There are actually, I can take one single type of molecule and make a bilayer membrane, and that would encase my cell contents with no problems whatsoever. But in cells, there are actually thousands of different molecules that form up those cell membranes, and we don't have a really good understanding of why there are so many, what they all do. And one of the reasons for that is that the membranes are really highly dynamic, so they're changing all the time, and we have no tools to see exactly which molecules are where. And so we're trying to develop tools that will allow us to track those processes, those movements of different lipids in a biological membrane.
Amazing. So, when you're building these sensing tools, is this using those labs on a chip that we've heard about in the news?
No, our sensors are all fluorescent sensors, so basically used in either fluorescence microscopy or flow cytometry to light up a particular species being present in a cell.
Right. So they bind with what you're looking for and they light up.
Yeah, absolutely. So yeah, the act of binding causes the fluorescence to turn on.
Right. And I guess that because so that's how it applies to I guess to waste you were saying, so they'd be able to bind to the things you want to remove from the water, for example.
Yes, absolutely. So exactly the same physical chemical interactions with the anions, and for pollution monitoring you might want the same kind of turning on response, so lighting up to say yes there is phosphate in this water, or no there's not. You might want it to change color. The little test strips that you use to monitor your pool water, it's the same kind of principle. If you could dip a test strip into a water stream and say the pollution levels in this are okay or they're way too high, that color change or that fluorescence change would be really valuable. The same types of molecules, but without the turning on of fluorescence or color change, can be used to bind the molecules and potentially extract them out of the waste, depending on, whether or not you've tailored the physical properties of the molecule to allow it to do that.
And so tailoring the properties of the molecule, that's what you're also learning from nature?
To some extent, yes. Yeah. So we look at how nature, what changes in natural molecules make them more or less selective for binding to whatever species they're binding to, but mostly for me, anions. But also then looking at how does nature use changes in molecular structure to change function?
Mm. And that's got all sorts of implications. So, supramolecular biology, if someone wanted to study and enter that field, what's the path? Because that seems very high up on the sort of molecular biology
Yeah. So,
ladder.
well, it's a supramolecular chemistry from my side of things, and it's trying to mimic biological processes, but it's basically the tools that we use every day are the tools that are used by organic chemists who synthesize new molecules, new molecular architectures, and by physical and analytical chemists who measure molecules. And that's really what we do. We make new molecular structures and then we measure their properties.
Amazing. So, you're creating new types of molecules all the time.
Yes.
And what's your main current focus? Because it sounds like you've worked on quite a variety of projects.
I have worked on a large variety of projects and continue to do so, I guess. We've got a couple of things running in parallel at the moment. So one of the things that we're looking at is how peptides and proteins that naturally bind to lipid membranes do that, and then trying to pull apart those interactions and build small molecule mimics to do exactly the same thing. And that's a really big focus of my group at the moment because of this lack of tools for membrane lipids. It's an area that it's a really challenging area. It's really hard to build these tools, but the impact of, if we can do it, will be huge because it will unravel lots of different biological processes. So that's one of the streams that we're really focused on. The other two, one is focusing on molecules that are created in cells as part of the Krebs cycle. This is a reason because whenever I talk to biomedical researchers and say, there's lots of negatively charged species inside cells, if you could have a sensor for one, which would it be? They always come back with these Krebs cycle intermediates. So things like citrate and succinate and fumarate.
Sorry, and
Which
the Krebs cycle is
it's a cellular energy production cycle.
right.
Yeah.
Sorry, and
So
you're saying what they look for.
yeah. So looking at it's a metabolic cycle, really. So looking at how cellular metabolism works and where interferences in that process might lead to disease. So buildups of some of these molecules in your cells is for example, a good marker of a cancer, a cell becoming cancerous. So one example of that is citrate, which is elevated in prostate cancer specifically. And so looking at, can we monitor the levels of that in cells that would allow us to diagnose quickly whether or not somebody is likely to have prostate cancer in a way that is similar to the current tests. The current tests use prostate-specific antigen to look at that, but that leads to a huge number of false positive results. And so whether this could be a viable alternative or not, to have something that I could dip a stick in some urine or some seminal fluid or whatever and say, That level's high, probably need to go to the next stage of diagnosis, or, yep, you're fine. Go back to doing whatever you were doing.
Like a rapid test.
Yeah, yeah, absolutely. So just a quick little dipstick in the GP surgery that wouldn't mean, you had to have, multiple blood samples taken or biopsies or anything like that. Um, so that's one of the other areas. So the sort of biomedical metabolic cycle intermediates, I guess. So again, looking at fluorescence sensors that could be used to monitor their levels in cells or in physiological fluids. And then the third thing that we're focused on is looking at sulfate binding. Sulfate is present in lots of water streams, whether that's feed streams. It blocks up pipes in the oil and gas industry because it causes precipitates that are, barium sulfate and calcium sulfate. Calcium sulfate is, that scale that you get on your kettle after a while, those sorts of things. And it's also present in lots of waste streams, so mining waste and nuclear waste, as it turns out. And in mining waste, it helps basically to keep some of the heavy metals in solution. So taking the sulfate out would be valuable in terms of getting those metals out of the waste stream. In nuclear waste, sulfate is a particular problem because it interferes with the process of turning that nuclear waste into a glass. So the whole sort of nuclear waste storage thing really relies on the waste being turned into a solid form that can be stored for centuries, millennia. And sulfate causes problems with that so that it makes the waste leaky, the solid leaky. So if you could move the sulfate out of the waste and do that without, taking all the radioactive stuff with it, then you would be able to process that waste much more easily and safely. And so that's one of the big applications on that side of things. So we're looking at trying to design molecules that can selectively bind to sulfate in nuclear waste and be able to then use them to extract the sulfate out of that waste safely and without, causing lots of radioactivity to get everywhere.
Yes. Such an important problem when we're going to be storing all this waste.
Well, yeah, I think it's going to become much more important over the next few decades, right? With the whole AUKUS nuclear submarine agreement. So something that I think is definitely worth doing. That one of the reasons that we got into this research area is that it's really well documented in the US. There's sort of this big waste site called Hanford, and they just don't know how to deal with the waste that they've got. They can't do anything with it. So, and drums of fluids get leaky all the time. They're full of corrosive chemicals. It's a big problem that I think we need to look at solving.
Oh, it's very good that you've got a handle on it. Is there anything I haven't asked you that you'd like to talk about?
Oh, let me think. um, Not particularly. I think
All
that,
right.
my research is quite wide ranging. I think one of the other things that I do like to talk about is the fact that it's not all about the research that we do. That's what, the research problems are really important, but training our next generation of scientists is something that I'm really passionate about. I work with teams of PhD students and early career researchers. In the field of chemistry, it's really common to have teams of 10, 20 people working on these problems. And it's why I can work on sort of a few different things at the same time. But the people who we train become the scientists of the future, and most of them are way smarter than me. I'm really lucky at the University of Sydney. We have some amazing students who come through. And so giving them the skills to be able to develop their own research into the future is something that I think is equally important to the research problems that I'm working on, and probably a more valuable contribution to society as a whole.
We do need more scientists.
Absolutely. And more people who understand science, who aren't just scientists, but who go into other areas as well, whether that's politicians who actually know what they're talking about, or just, the people in the street who need to understand science to understand, the way the world around them is changing with things like climate change. Yeah, I think it's
Absolutely.
really important.
I
Yeah,
think I think politicians can't really develop a sensible policy unless they understand a lot of the science around the areas that they're trying to develop policy for, and then people don't know how to vote for it if they don't have a
if they
general
don't.
understanding as well.
Absolutely, absolutely. So yeah, I think that, training scientists, but training people in science who then move on to other areas in their careers is one of the most important things that I do. Absolutely.
Mm. Um, in that case, would you like to make any concluding statements, or did you just do that?
I think I just did that. Science is fun.
Science is fun. Do more science.
Do more
Well,
science. Yeah.
well, Kate, thank you very much.
No worries, Ian. It was lovely to talk to you.
Study science, because you will find in the study of science a richer, more rewarding life.